Thank you so much for coming virtually. I really appreciate you coming to support our Sea Grant interns to the Michigan Sea Grant program this summer. We're really excited to hear more about their projects, and we appreciate everyone who supported their work this summer and made what they were doing possible, from their mentors to their families to Mike Fraker's family. So thank you all so much. We as Michigan Sea Grant we call ourselves a three-legged stool, where we fund research, education, and outreach projects and work throughout the state, where our goal is to foster science-based decisions about stewardships of Great Lakes resources. We consider the Great Lakes to be one of our most precious natural resources in this region, and we are all, we've dedicated our careers to helping with that. I also want to point out that although we are at University of Michigan today, Sea Grant is a cooperative program between University of Michigan and Michigan State University. And a lot of our partners and partnerships are through our Michigan State Sea Grant Extension program. So, and we even have someone from Extension here today. Thanks for joining us. We are committed to diverse and inclusive engagement to facilitate research, education, and partnerships. And I think that those partnerships are really highlighted here today through the various mentors and organizations that have worked with us to support our interns this summer. And we encourage anyone else and the rest of our partners across Michigan to consider supporting an intern next year. We fund the interns, and the mentors provide often an in-kind match of support for them. We coordinate and fund undergraduate students, and they work with local government, state government, federal agencies, nonprofits, sometimes private businesses, sometimes university faculty, as long as it's in a partnership with an external partner within the state of Michigan. But the idea is to really give students a hands-on experience of the kind of thing that is available to them, whether it's stewardship, education, or fostering management of the Great Lakes. And we hope that they will move forward in their careers to continue to support those values. We are in part funded by a fund that our former colleague, Mike Fraker's family, helped set up last year. Mike was really instrumental in getting this program set up. It is the biggest and longest running internship program in the entire National Sea Grant program. And Mike was a huge part in getting it set up, and he shepherded the original interns through the program. And so we are really excited to continue that legacy and have the evidence of that before us today with this year's crop of interns. And we're really grateful to everyone who has continued to support the Mike Fraker Fund. And we are continuing to fundraise so that we can keep doing this in the future. We will be sending you interns a Google form. Please answer the questions. It's really important for our reporting, but also because even though it is the longest running, we haven't actually been doing it that long. And so we'd be happy to have feedback from you on how to do it. Particularly, we may need to sort of rethink the beginning part of the process, where you're finding partners or whether partners are giving you projects. So if you have any thoughts for us on how well that worked for you or didn't work for you, we'd really appreciate the input.
Hi, everyone. Thank you for being here. My name is Ella, and I am a fifth-year undergraduate student studying environmental science at Wayne State University. And today we'll talk about my Sea Grant project, which was to help the Clinton River Watershed Council advance their Stream Leaders program. And we'll talk all about that. But first, just a little overview. First, I'll just go over CRWC and what the organization does. I'll talk a little bit about Stream Leaders and outdoor education in general, my project specifically, and some foreseen impacts, a little discussion, and then we'll wrap up. So a little bit about CRWC. We are located in Rochester Hills. We are a nonprofit that was established in 1972, and we host community events, resource access, and programs like Stream Leaders. And all of our work is guided by 4 pillars to protect, enhance, celebrate, and engage the Clinton River watershed. And if you are unfamiliar with what a watershed is, it is, as you can see, an area of land where water naturally collects and drains to one point. And in our watershed, the water is in the streams and the tributaries, and that eventually goes into the Clinton River, then Lake St. Clair, the Detroit River, and then the Great Lakes. And our watershed is the most populous in Michigan with 71 communities and 5 counties. Okay, so about Stream Leaders, this program is a place-based outdoor educational program, and it is available for upper elementary to college students. And the 2 goals of the program are to raise awareness of the importance of local water quality and for students to create connections to the Clinton River watershed. And the program does this by providing 3 monitoring stations during the program, which are biological, physical, and chemical. And this is where the students, they get hands-on real-life monitoring experience. And each station is led by an intern or a CRWC staff member or any other mentor. And the students have their own worksheets that they complete during the monitoring process. So just to briefly go over each station, the biological station is where the students, they get their waders on and they get into the water and collect benthic macroinvertebrates. And you could also call those aquatic insects. And the students do this using either a D-net or a kick net. At our council, we usually use a D-net. And you can see a video of students demonstrating it here. And when the students collect the bugs, they ID them using an ID key to overall find a total stream quality score. And the score is found depending on the different kinds of bugs and the number of bugs that they find, because they can be divided into 3 different categories, from most pollution sensitive to least pollution sensitive. So it just depends on how many and which bugs you find to get the score. And it ranges on a scale of 1 to 4, which is poor to excellent. And these are just some examples of the aquatic insects we can find in our watershed. And this is just a snippet of the worksheet where we can calculate our score. And for the physical station, this is where the students, they physically assess the water body that they are monitoring. And this can be done through seeing what odor the water has, what color is it, is there any vegetation around, what kind of vegetation is it, is there any animals around, what kind of animals. And they also measure the velocity of the stream and the width and the depth. And you can see some students here measuring the velocity. And then here's another snippet of the worksheet. And the last station is the chemical station. And this is where the students measure multiple chemical factors of the water. And this could be fecal chloroform, pH, turbidity, and more. And they do this using Lamont chemical testing kits. And they have very easy to follow instructions for the students. They have chemical tablets and test tubes for them to collect the water in. And depending on what the results are for each test, it helps determine an overall water quality score, also on a scale of 1 to 4. Here's the worksheet again. So once all the Stream Leader sessions are done, and an important question to ask is, where does this data go? And it's a good question. So it goes on the Clinton River Watershed's website, and it is available for public view. And this map is from 2021, and it has monitoring data from 2021, and it was recently updated for my project, which we'll talk about. But yeah, this map is interactive and available to the general public. So talking about Stream Leaders and outdoor education, why is outdoor education important? Why do we have a program like Stream Leaders? Why does it exist? And outdoor education provides multiple benefits to students who participate, like social and personal development benefits, like teamwork, leadership, enhanced self-confidence, and enhanced decision-making skills, and pre-academic skills. It enhances cognitive, linguistic, and motor skills, and it also benefits mental health, like decreasing anxiety and increasing life satisfaction. And we know that Stream Leaders directly connect students to their watershed through the monitoring they are doing. And we know from research that connections to nature can foster environmental stewardship. And according to the Great Lakes Stewardship Initiative, place-based learning increases civic engagement and responsibility. So all of these are being created by Stream Leaders and outdoor education itself. So to give some impacts of Stream Leaders itself, now that we know outdoor education impacts in general, we know that Stream Leaders has reached over 70,000 students overall since its existence. which is a lot. And Student Congress is another impact, which is after students monitor, they go to a professional gathering where they can share their results and come up with a community action plan. And depending on the class, this may or may not be fully developed or implemented yet, but it's taking that stewardship portion of Stream Leaders and putting it into play. And it is currently, Student Congress is currently not active, but we're hoping to get it back up soon. So talking specifically about my project, there were many different things that I had done to help the overall goal of program advancement. And the first step was mentorship. And that is where I learned the ins and outs of Stream Leaders itself. And I acted as a mentor for some of the stations. And I really learned from my fellow interns what to say to the kids when they come to each station. And so it was a really great way to learn about Stream Leaders. And so also during this portion, I learned, identified some program areas that could be improved, like safety precautions, like always reminding the kids to wash their hands after monitoring in the river or to wear gloves. And just thinking about some location changes that could occur for better monitoring. Another portion was research, and this included business mentorship and researching schools. And so for businesses, CRWC staff can't always be at every monitoring session. So it's extremely helpful when businesses like General Motors, they can learn how to do Stream Leader sessions and collect their own data and then send that back to CRWC. And unfortunately, during the COVID-19 pandemic, a lot of our mentorship was lost. So my goal and my task was to research local businesses within the watershed that I thought could be potential volunteers for us. And I identified many automotive ones and overall 48 businesses. So this is just a resource that we have to reach out to businesses to see if they want to be involved in collecting data for us. And then for the schools, this was just researching schools within our watershed that we could reach out to see if they want to be interested in participating in Stream Leaders. And so far I've identified Macomb County schools, and I started Oakland County schools, and I heavily utilize the Michigan Intermediate School District guide to help me with this. So again, another resource that we can tap into when looking for more sessions. Another portion was the recruitment toolkit, and this is, in a nutshell, a guide for teachers to know the who, what, when, where, why, and everything in between of Stream Leaders. And it's just a streamlined way for them to know everything about the program, like program information, the process timeline, benefits, etc. But one of the most important portions of the toolkit would be to show the teachers the Michigan K-12 science curriculum connections to the Stream Leaders program. And it would show the teachers, hey, this program is not just kids playing in water and collecting bugs for fun. It is fun, but it has a purpose and it has learning objectives with it. And this you see here is the outline of the toolkit. It hasn't been created yet, but it's in the works and it was started. And lastly, maybe the most important part was data input and mapping. And thinking back to the map that we saw earlier of the 2021 data and how it was updated, this is that portion. So here on the left, you see an archive of Stream Leaders monitoring locations and schools all the way back to 1996. And so this data was taken and put into Google My Maps. And from there, it was put into ArcGIS Online, which is a mapping program if you're not familiar. And 2022 to 2024 monitoring data was put in there as well. And so this map will soon be available for the general public to view and to click through, and you can remove layers and add layers. So lots of more information available. So I have obviously not seen the impacts of my project yet, but some impacts that I foresee happening are from our mapping that had occurred on Google My Maps and ArcGIS, just the council being able to identify where we have not reached within the watershed for programs, and finding areas within the watershed that might be underserved by environmental organizations, excuse me, and just seeing how we can get those schools access, whether that might be through busing or providing other funds to get them access to the program. And another impact could be the program data being available to the public. And this could just show the general public, hey, the watershed cares about this. Maybe you should care about it too. That might be a rose-colored glasses view, but it just shows them like we're doing this work and it's important to care about water quality. Teachers can use this data and compare their own monitoring data to other stream leader schools and maybe create a lesson about water quality using the data. And then maybe another rose-colored glasses view is policymakers taking a look at it and getting involved with water quality and seeing that, hey, we care about it. Maybe you should care about it too. Not maybe, they should. So, and then another one is mentor search and toolkit creation, and just recognizing that like a ripple effect, more mentors equals more sessions, more connections between the students and their watershed, or just the businesses in the watershed, and just more stewardship being created through these sessions. So that was my project, but my main focus, my main focus besides Stream Leaders was helping out with some side events that I really enjoyed and had lots of fun at the watershed. And one of my favorite events was red swamp crayfish trapping. And if you did not know, they are invasive in Michigan. We did not find any, but it was really cool to do. And then just collecting bugs for outside events just to show kids the bugs. And then drone surveys over Lake St. Clair for looking for woody debris. So really cool. And just to emphasize some points, if you could take away anything from my presentation, I would say that not all classrooms have 4 walls. And I think that is the overall goal of Stream Leaders and outdoor education itself. And just remembering the benefits that it has for students and how it can foster environmental stewardship and pro-environmental behaviors in the students that are involved. The toolkit development and mentorship, and just the cultivation of more leaders and schools involved in the program, and just getting more sessions and more connections, more stewardship at the end, and then program growth and accessibility, just providing more access to the program and finding ways that we can do that. Thank you. I would really like to thank the Michigan Sea Grant for this opportunity. This is my first internship ever, and I learned a lot, and thank you. And so for Haley and everybody at Michigan Sea Grant, thank you. And also to the CRWC staff, Kaylee, Jennifer, Pranoy, my supervisors, thank you. And my email's here, you can always email me, and feel free to find me after. I have LinkedIn. I'd love to connect with you and talk to you, so thank you. And do you have any questions?
Thank you. I actually have 2 areas of questions, so to keep you from running back and forth.
Yeah.
2.
Yeah.
One, being a former teacher, what about the students who were involved? Was it a requirement from a course? Was it you accepted anybody who was expressed interest, or was there any kind of a screening process?
Kaylee is my supervisor. She's here. That'd be a really good question for her to answer.
Yeah, so for the schools that participate in the program, the teachers make it optional if you want to opt out of the field trip. Otherwise, it's definitely encouraged and the majority of the class attends. But you know, if a parent isn't comfortable or something like that, it's like signing a field trip slip.
And the other question was about the businesses that you were trying to involve. In general, were you encouraged or discouraged by the response? And did you, in approaching those businesses, did they have like an identifiable person, like somebody for health and safety, or was it the communications person, or did you go to some other corporate office to try and initially get your entree?
That's a really good question. So I actually didn't reach out to businesses. I just compiled a list for us to reach out to in the future. But I actually had a difficult time trying to find a specific person to contact for volunteer opportunities. So a lot of the emails and phone numbers that I put were just general numbers. So yeah, to answer your question, it was a little difficult to find the businesses, like a general contact. So yeah.
I'm sorry. If I could make a suggestion for the future, then what you might be able to do is to communicate with either the HR department or corporate communications and explain your purpose and ask them who is it within the organization that we could directly contact.
Yeah, that's a good suggestion. Thank you.
Well, thank you. That was a great presentation and it's a really, really exciting project. For those of you listening online, this is Sylvia. You heard from me earlier doing the intro. And I just wanted to point out that this is in the context of something that's getting a lot of regional importance and emphasis right now, the International Joint Commission, which is a body that's overseen by the government of the US and the provincial governments that border Canada. It makes policy and other kinds of recommendations to Congress and Canadian governments. One of the big pushes right now is for what we used to call citizen science and are now calling participatory science programs, and developing a sort of regional hub that coordinates programs that are already doing this very successfully, and trying to make better resources and funding and like a more permanent program for this within the Great Lakes region, in a way that those data that you work so hard to get online would be in a sort of regional repository and get used by managers, and have go through like a QA/QC process. And we have 3 fellows right now that are working with the IJC to come up with help with the implementation strategy for this. One of them sitting in the front row, Ian. You might want to talk to him afterward. But I just want to say it's super exciting that you're doing this. And at the time that we selected your project, we just did it because we thought you were great and the project was great. But seeing how it really fits into this bigger picture is really exciting. So I encourage you both to follow up with Ian and talk about ways that you all can participate in the IJC process.
Thank you.
Question from me, what are some skills or lessons learned that you're hoping you can take from this forward into your career eventually?
Yeah, one of them was field work, and I had never participated in field work before this, and just learning the procedures of field work, like how we did for red swamp crayfish. I really enjoyed that, and just learning processes of how to collect insects and do field work in general. I think that was a big skill. I think rekindling my communication skills with kids was a good one. I don't know if I'd take that into the future, but I think that was a skill I gained. And meeting environmental professionals at any events that I went to with Kaylee and for the council in general. So those are some skills I think I gained.
All right, my name is Clay Wilton, and for my project, I did acoustic monitoring of bats in northern Michigan. So a quick overview. I'm going to give you an introduction to myself and to the background of this project. I'm going to go over some of the objectives for my project and the design of the project. Then I'm going to go over some of the data analysis steps that I've been taking and give you some of the results from the data that we have in so far and some conclusions from that. And then I'll give some brief acknowledgements for organizations and individuals that helped me along the way. That's me. I'm Clay, as I mentioned previously. I was raised in Dansville, Michigan. I graduated from Dansville High School in summer of 2021, and I'm currently attending Lake Superior State University. I'm going into my senior year, expected to graduate spring of 25, majoring in fisheries and wildlife management. So a little bit of background on why I'm doing this project. So what you see here is a bat that has been infected with a disease known as white nose syndrome. Now, white nose syndrome is a fungal disease that infects grows on the skin of bats, and basically it eats away at them, sapping their energy. And during hibernation is when it's most lethal because they are not getting energy back in. It's forcing them to wake up and forage at times they're not supposed to be doing that, and it can be and most often is very lethal. It arrived in New York, so it is an invasive disease, in 2006 and made its way to Michigan around 2014. And since then, it's been spreading westward and it's been leaving a trail of millions of dead bats behind in its wake. And we know it's in Michigan and it's been through most of the state already. And really what we're looking to do is damage control, seeing what we have left, what we can do to help restore damaged bat populations. So for this project, we're looking at what species that we have left and where are they and where are the best areas we can focus conservation efforts for our remaining species. And like I just gave those objectives, but you know, here we are. But so more specifically, we're looking to do some species richness analysis, number of species in each given area, what species are they? And hopefully find some habitat preferences so we can focus conservation efforts on specific habitat types, focus down our efforts a little bit, and make it easier for wildlife managers to help these bats recover. And I didn't really know what to put for pictures on these slides, so I put up a picture of a little brown bat and a silver-haired bat right there, just because I thought they looked pretty cute. I don't know about you guys. But for our actual experiment, we have several sampling sites. We have data for 15 of them, but there are more in the works. These sites are located along coastal areas of the northern Upper Peninsula or eastern Upper Peninsula and northern Lower Peninsula. And these sites fall within the boundaries of the U.S. Fish and Wildlife Service's coastal program. U.S. Fish and Wildlife is the partner organization that I'm working with. My supervisor is an employee of the U.S. Fish and Wildlife Services. And the site ownership varies from site to site, but most of our sites that I deployed to and that my supervisor deployed to fall under the ownership of the Little Traverse Conservancy, which is a land management, a land trust basically that's in that area. For equipment, we used these SM4 bat recorders, and in conjunction with these SMM U2 microphones that you see at the top of that pole there, they can pick up the frequency, the sounds on frequencies that the bats are using to echolocate to move around their environment and hunt for prey. And generally, these calls are distinct enough between species that you can run these through a data analysis program known as Kaleidoscope Pro, and it can tell you which bat species is making each sound, thus giving you an ID for different species within the area. So each of these recorders was set out for a period of between 1 and 3 weeks, just to make sure we had enough data to really make sure we weren't missing anything, you know? So after that, we are moved to our data analysis phase, which is still in progress, but I do have some results already. But this up here in the top is the a spectrogram from the Kaleidoscope Pro software. And you can see that these spikes there, those are calls from the bat species, and I have one from silver-haired bats up top and one from a little brown bat down at the bottom. So we're going to be running statistical analysis between the different habitat types and the different bat species that were present in each type to basically associate a species preference by site. And then I'll be mapping that in ArcGIS Pro to basically create a reference document essentially for future wildlife managers to compare to bat species in their area. And so some preliminary results we got, and I've got 15 sites here. These top, what is that? 8 sites. I and my supervisor deployed to personally. The remaining 7 at the bottom here are from a partner organization known as the Lake Chanot Watershed Council. And we found a few interesting things. So one of the more concerning things that we found was that in the Sault Ste. Marie area here, which is these 3 sites, we found no northern long-eared bat presence, which is concerning because they were known to be in that area beforehand. And while they are present in other sites, so they're not extinct or anything, we are worried about their existence throughout their whole range. But on the bright side, what we did find is that every bat species that was present in the eastern Upper Peninsula and northern Lower is still there, at least in some capacity. We particularly found that there's higher bat activity in the Lake Chanel area, but there was relatively low activity in Sault Ste. Marie, relatively high in the Lake Chanel area. So we might want to consider focusing our efforts towards the Sault Ste. Marie area, and less so on the Lake Chanel area, but they are both important. One thing I didn't mention earlier was that in a study done I believe this was a 2021 study, the Western Upper Peninsula was found to have lost 89.9% of its bats in total. And given the similar makeup of the similar habitat rather of the Eastern Upper Peninsula, we are concerned that we would find similar numbers. And one thing I'm seeing here is that while these numbers look pretty good, apart from the northern long-eared bats, they're nowhere near what we really would have found before white nose syndrome came through, which is as expected, of course, but still unfortunate. But there is still hope because we do still have bats in the area. And to wrap things up a little bit with some conclusions, like I mentioned, we had sites all over the Eastern Upper Peninsula, and we have some in the Northern Lower, but we just don't have data for them yet. So right here, we've got the Sault Ste. Marie sites that I was mentioning earlier, and down here are these Lake Chanel sites, and we had a few out in Drummond Island that we got results for. What you're seeing in the bottom left here is what I'm currently working on, that land cover analysis that's mapping the different forest types, and we're hoping to associate those with different bat species presence. And this is all being done in ArcGIS Pro. So that's currently being worked on. But yeah, that's pretty much my whole project. I'd like to give some shout outs to Danielle Fegan, who is my supervisor on this project with U.S. Fish and Wildlife, and to Dr. Jill Witt, who is my LSSU my student mentor for my upcoming senior project that I will be expanding on the work that I've done in this project to complete. And of course, a big thank you to Michigan Sea Grant for making this all possible. I wouldn't have been able to do it without the funding from you guys. And of course, thank you to Lake Superior State University for giving me some of the resources that I needed for this project, and to Little Traverse Conservancy for graciously offering us to be able to use their properties to do this monitoring. And a thank you to U.S. Fish and Wildlife Service for providing the recorders and the microphones that were used in this project, and to the Lake Chanel Watershed Council for sharing their data with us. Were there any questions?
What do you see as the long-term impact if those bats do not reappear? Environmental, I don't know anything about bats, but is it their food source that would then become uncontrollable, or what other environmental impacts would there be? Thank you.
So one thing that I've noticed personally is bats, by the way, in case you aren't already aware, prey, the ones that we have in our area prey on insects. And we have a large amount of insects currently, I believe partially attributable to the distinct lack of bats in the area. Bats are also known to be worth about $3 billion to U.S. agriculture for like essentially free pest control. And some bats do pollinate. Not in our area, but there are others that outside of our area that are still susceptible to white nose syndrome that do pollinate plants.
So great presentation. I'm curious, one about, are there other factors that you're looking at for the bat decline, or is it white nose syndrome? So it's called white nose syndrome.
It is
the only factor or the primary factor?
It is by far the primary factor. Bat species populations have been pretty stable until white nose came along. It has been extremely devastating, and it's really the main thing that we're focusing on. Of course, expanding human habitation is always a concern, but bats do like to roost in attics if you let them. So it's not necessarily the worst thing if they're less affected by human development than many other species are. Not to say that they're unaffected, but no, white nose syndrome is the main factor that we're looking at here.
Thank you so much for that presentation. I did want to point out bats eat like 1,000 mosquitoes a day.
So they
So
can eat
great
well
for
above
pest
their
control.
body weight.
I was hoping you could talk to us a little bit more about your partnership, like your faculty mentor, and then you're working with U.S. Fish and Wildlife Service. Can you talk a little bit more about that interaction and if you received mentoring, or was it just like the providing of the equipment, data sharing, that kind of thing?
Yeah, so U.S. Fish and Wildlife Services is my partner organization, as I mentioned. My supervisor was very, very helpful with this project. I couldn't have done it without her. She showed me a lot about field techniques. U.S. Fish and Wildlife provided the equipment that we use, but also in the form of expertise to be able to get this project done. The data that we are collecting is actually going to be shared on the North American Bat Monitoring portal, which is available to the public. I don't believe we've posted our data yet, but it will be available in the future. North American Bat is basically a database that will show, where bats are. It's an active monitoring and record keeping source. It's very helpful for land managers and conservationists looking to help bat populations in the area. Some takeaways from this project on my end are I got to learn a lot of new field techniques. I got to do some networking with some local agencies. It was just a really, really good experience for me. This is also my first time getting out into the field and doing this kind of work. And it's just a really great opportunity for me, and I hope it's going to be a really good resource for conservationists in the future.
So 90% devastation since 2006. Is that my understanding is correct?
Yeah, so the white nose syndrome arrived in 2006 in New York State and spread westward since then, arriving in Michigan in 2014. And it's been devastating ever since it landed.
So what are some of the next steps? Like, you're monitoring their activity, but what are some of the things I would assume that are in place or going to be put in place to, to help mitigate this, to stop it? Because it's so devastating. I'm surprised it hasn't gotten more attention, actually.
Yeah, so there are a few things that we can do. One of the things that's being done on a larger scale is they are developing a white nose syndrome vaccine, which is in field trials. It's especially going to be helpful for areas that haven't had white nose syndrome come through to inoculate bats before they can get sick, infected with the disease. In areas like ours that have already had it come through, we really just need to be focusing on making sure the bats have habitat to recover in. I did read a paper that suggested using UV lights to attract insects around different known bat habitat to allow them to recover energy, especially post-hibernation when they're most vulnerable. The bats that do survive will need to have ready available food sources to replenish their depleted energy and depleted fat reserves. So these UV lights will attract their preferred insect prey to one place, and that will really help bats recover post-hibernation.
Thanks. Also, one last question is just how far along is the vaccine? Do you know anything more about that?
So I know it's in field trials and there are certain numbers. I'd have to look back over some of the results, but I do know it's in a pretty good spot right now. And I believe they are going to start rolling it out not too far in the future if they haven't started already.
Okay, so I guess I'll get started here on my presentation. My name is Abraham Stone. I'm a student at Michigan Technological University, and today I'll be talking about the connections between plants and rocks on the Keweenaw Peninsula shorelines, which we've termed the Rocks and Roots Project. And so first, I'd like to get started just by acknowledging everyone that I'm working with. I'm a biologist. I love working with anything that's living, but especially plants and fungi. And that's been the center of my education for the past 4 years. And I happen to work with a bunch of non-biologists, which has been super fun for this project, primarily Dr. Erica Vai and Daniela Zadra McPherson, who work at the Great Lakes Research Center at Michigan Tech, and they are geologists. And I've learned so much about rocks from them over the past several weeks. So my sponsor that I've been working with for the past several weeks has been the Keweenaw Community Forest Company, which is a local nonprofit up in the Keweenaw Peninsula. Works a lot with conservation and making sure that land stewardship is the #1 priority for a lot of Keweenaw land. I'd like to extend an additional thank you to Janet Marr, Keweenaw botanist extraordinaire, Adam Bastian, my field assistant, and Carol Reschke, plant community ecologist, for helping design the introductory parts of our project. And then through the Keweenaw Community Forest Company, Gina Nicholas, its president, who I've been able to work closely with, and then Molly Powers, wildlife ecologist and land steward, who has joined us for a lot of our outdoor explorations. And of course, the Michigan Sea Grant for being able to stimulate the connections that we've been making on the Keweenaw and kickstarting this epic project that we've been doing. All right. So I want to start here by asking you guys a question. We are all here because we love our shorelines, and all of us are here because we're part of Michigan Sea Grant. We've been working closely to the shores at the connections between land and animals and water. And I wanted to ask you on a personal level, what connects you with your shorelines? What brings you to the spots that you always visit? Is it the birds? Is it the sounds? Is it the views? There's so much to love about our lake shores. And I live on the Keweenaw Peninsula, and so I'm surrounded to the north, to the south, to the east, and the west by shoreline. And so here's a couple of the things that I care a lot about. On the top left, that's one of my favorite flowers that we see on the Keweenaw Peninsula. You can find it in the Straits of Mackinac too. It's called a Mystacini primrose. It's a really special Great Lakes plant. It's an Arctic plant that makes its way down to the Lake Superior shoreline. And it's one of many beautiful plants that you can find on the shores. The rocks and the lichens are also beautiful. They're exciting to see, and there's so many different types of them across our shorelines. It's also the best spots in the world in the United States for Northern Lights viewing. That's a picture I took last year. And it's beautiful all times of year, winter, spring, summer, or fall. There's always something to see. It's also, if you've been to the UP, on a hot day with a south wind, it's also one of the best places to experience black flies, which I can still hear in the back of my head. And those shorelines are so diverse. We have, especially on the Keweenaw Peninsula, we have cobble shorelines, and we have bedrock shorelines, and we have sands, and we have every type that you can imagine of substrate along these shores. And there's so much to be into and so much to explore, and you guys all connect with in your own ways with these places. And today I'm going to be talking about a couple of them that I've been looking at more closely. And those are the plants, the rocks, and how they change across the course of our shorelines. And so I'm going to begin by talking about the rocks a little bit, talking about the geology of the Keweenaw and why it's so special. So let's go back a billion years ago, almost a billion years ago. We were part of a giant supercontinent. And right in the middle of that continent, it was starting to split apart. It was a rift valley, which you guys might know about the Great African Rift Valley. That is probably the best analogous area to what happened here all those years ago. And so the continent was being pulled apart. And as the continent was being pulled apart, it started to have the softening of the crust. It started to get thinner and thinner. And so the pressure was being released for all the magma and that was up in the mantle. And so we started to get these volcanic eruptions that would come up in the Rift Valley, and you get these slow moving bodies of lava that would make these large lava lakes. And these became known as as the Keweenaw Fault volcanics. And so you had, as the pressure started to get released, you had more lava moving up, and it made its way to the surface, and then it cooled after becoming a lava lake. And some sediment came on and, you know, deposited a layer of sandstone or a bunch of boulders on top. Then you get another lava flow, and then you get another cooling, and then you get more lava, and then you get more cooling and another sedimentary layer. And so you started to get these alternating layers of lava flows over the course of millions of years here. And eventually the rift stopped being super active. It started to cool down volcanically. And so you get then this large layer of sediment that's, you know, several, you know, however thick that next layer was, but it then got capped. And so we call this overall volcanic thing, not just the Keweenaw Fault or the Keweenaw Fault volcanics, but the Portage Lake volcanics as they're found in the Keweenaw Peninsula. And it's present across much of the United States and a little bit of Canada there. This is all where those volcanics took place, but they're most prominently on the surface here in the Keweenaw Peninsula. And as you can imagine, with all of these flows and all these layers of sediment that we're depositing on it, we get lots of really, really cool rocks that grow here. And so all of these are present here on the lakeshore, and they're present in different areas. And to add real quickly, they make up the Keweenaw Peninsula here, but they also make up all the rocks on Isle Royal, because right in the middle between them and Lake Superior is a syncline. And so that's where the water accumulates. So that rock that's under here, this is all sedimentary rock that came from the last, you know, after those, you know, millions of years that you had those volcanics, everything that came after, that's what you see here. But it makes this exposure here at Isle Royal and on the Keweenaw Peninsula. And so they're actually the same rocks. And so on the Keweenaw Peninsula, we have several different types of bedrock that we can find here, and they're super prominent along the shorelines where there's lots of erosional processes. And so those alternating layers that I was talking about, those became known as the Portage Lake volcanics. And those are present mostly on the southern part and the way western or eastern part of the Keweenaw Peninsula. That's where they make their exposures. And within those, you also get these giant blobs of lava and magma that cooled and formed these huge batholiths. And those became known as a different type of rock called rhyolite. And that's a much more complex process, but they also make their appearances on the southern part of the Keweenaw, especially down here in an area called Fish Cove. On the northern side, like I mentioned, everything got capped after this volcanic period was done erupting. That became known as the Copper Harbor Conglomerate. And that occurs on the way northern tip of the Keweenaw Peninsula and makes up that northern shore. It's like the iconic Keweenaw shoreline is those really harsh red conglomerate rocks. And sometimes after that, all that conglomerate came in, you had still these little spurts of activity. You had these little bits of lava that just somehow made their way through the conglomerate and then created a new layer on top. And those became known as the lakeshore traps. And so on the North Shore there, you sometimes get these interesting isolated areas of basalt that came up many, many years after the initial Portage Lake volcanics. So as you can see, we have there's here geologically, and many of these lava flows, there are lava flows all over the world. It happens, but here are some of the most special ones in the world because these are some of the deepest lava flows that have ever occurred. The Greenstone Flow, which makes up the backbone of Isle Royal, which you also find on the Keweenaw Peninsula, is almost 400 feet thick, which that's twice the depth, almost twice the depth of Lake Erie. I mean, imagine a lava flow that deep and it was on the surface of the earth. So these are crazy, crazy formations that we get to see now almost 1 billion years later. And amazing things grow on top of them. Because here we also have some really special relationships with our plants. They're really cool plants that grow exclusively on the Keweenaw Peninsula and then the nearby area. And there are two main types of them that grow here. You have something called the Arctic relics, which are plants that make their way down from the Arctic after the last glaciation, after all the glaciers were moving back up north. You had a couple areas where they stopped because there was a large body of water and these plants were kind of left to just grow. They couldn't move any farther north. And they've somehow been able to live here in the Lake Superior Basin for the last couple thousand years, despite the fact that they're Arctic plants. They love cold weather. They're able to survive because of Lake Superior. So we get a couple of those plants here, and then we get a couple other different kind of plants called Pacific Northwest plants. And the historical relationships with these are a little bit murky, but these plants somehow grow all over the Pacific Northwest and into the South and into the Rockies. And then sometimes they grow in the Black Hills regions too, but then you don't see them for hundreds of miles until you get to the Lake Superior area. And so this plant right here, this is called the heartleaf arnica. This is a state endangered wildflower. And this is found exclusively in the entire eastern United States, on the Keweenaw Peninsula, a small population on Isle Royale, and then in the Thunder Bay region of Ontario. And it is hundreds of miles away from where all the rest of its population grows. And so, as you can see here, this yellow indicates it's endangered, but you can see that disjunction. It's pretty striking. So we're wondering, why do these plants grow here? And not only why they grow here, but why they persist here. And they likely have something to do with the bedrock here, because it's so unique, and our formation along the lake probably created an environment suitable for this type of plant disjunction. And so, this kind of abstract plant distributions and, climate history, et cetera, but these are plants that we connect with on a daily basis. These plants have really interesting migratory histories. So the Pacific Northwest disjuncts, have you guys heard of bilberries before? Is that something you've heard of? Okay, that's a UP, I think it's kind of a UP special term. It's one of our terms for huckleberries. It's for blueberries that don't have the little triangles at the base of them. And we have 2 of them in the UP, specifically really prominent on the Keweenaw Peninsula that we find. And both of them have this type of distribution again, that Pacific Northwest distribution. And so they're really special to the UP, but pretty much only to the UP in the Midwest. But they hold that cultural significance. And then the other important one that you've probably heard of, if you have been to the UP, especially Keweenaw, is the thimbleberry. And thimbleberries are another one of those Pacific Northwest plants, and we eat them all the time. They're delicious. And then we have a whole bunch of Arctic plants here, which I've taken pictures of. These are ones that I found along the shorelines. All of these like to grow here. Pinguicula right here, also known as the butterwort, is a carnivorous plant that grows on Lake Superior. And this one really likes calcareous soils. And it grows immediately on top of the bedrock, which means it really likes to have alkaline waters, alkaline soils, and without much of that dirt substrate, it must have some type of intimate connection with that rock in which it's able to grow. So we want to parse these relationships out a little bit. And this ecosystem that I'm talking about, the connections between the geology and the life on top of it, has already been recognized by the state of Michigan as a volcanic bedrock lakeshore ecotype. And it's imperiled in Michigan because it's so limited in its distribution and because it's so susceptible to development. Obviously, it's in one of the most beautiful places in the state, so it's going to be very lucrative for people to sell it and build houses, et cetera, et cetera. And there have been some relationships seen between the plants and the rocks that they grow on top of. In the 1990s, there was a very large review survey done of these lake shores, and they noted that some types of rocks like conglomerate and vesicular basalt, which is basalt with lots of little holes in it, tend to hold more diversity than some of the other types of bedrock. But that has never been quantified. So what we want to do is try and figure out a way to quantify that, and we have to do that by becoming well-versed within the ecosystem. And so we have this idea, this hypothesis that we're working with, looking at all the ways that rocks can influence the type of plants that grow on top of them. And they range from geochemistry. What types of minerals are available in the soil that will allow specific types of rare plants and protected plants to grow there? Do different types of bedrocks erode differently? Are there positions on the Keweenaw going to influence what types of microhabitats can be formed? Because different positions influence the types of waves and wind action that we see on these shorelines. And then does the actual structure of the rock itself in very specific geology terms, would all of those things also affect the ways that plants grow on top of them? Strike and dip, and then aspect and slope, which way the rock is facing, and then how large is its extent? And so this summer has been all about this type of exploratory work, figuring out not only is there a way to survey this, but can we survey this across the entirety of the Keweenaw Peninsula? And for this, we've basically divided our work into two separate types of field work. First one is practicing a survey methodology that we can use, that we can put onto a conglomerate shoreline, put onto a southern basalt shoreline, put onto a cobble shoreline, and be able to standardize our methodology so that we can compare the different types of life on the bedrock, between types of bedrocks and within bedrock types. And so we did this by practicing with 6 different transects across multiple different bedrocks, the rhyolites and the conglomerates and the basalts. And we did so while also separating microhabitat lines. And so this is an example of the type of microhabitats we would look at. See this much darker spot along the shorelines that appears both here on the basalt and then here on the conglomerate? That's a very specific lichen that grows on there. And it has a pretty clear line where it stops growing and then is taken over by a much higher lichen diversity zone. And those microhabitats we call the splash zone and then the lichen zone. And so that's a natural ecotone that we can look at to see what plants really like growing in the splash zone. There's some of them that will only grow there, sometimes only in super exposed areas, sometimes in really protected areas. And some plants only grow in the lichen zone because it's much less disturbed and you get more diversity. It tends to have a larger area on the shoreline. So understanding those relationships has been key to our work and taking lots of notes. And then, of course, doing our practice methodology on them. Second section, which would be comparing our practice transects to other shorelines, not only looking at where we've practiced on, but walking along the entirety of the Keweenaw Peninsula shoreline to see if we are not incorporating every type of possible shoreline into our survey. Saying, can we take our methodology and bring it to a cliff and practice it on a cliff? Can we take it to a sandy beach on the southern side of the shoreline and see if we can figure out what plants are on there and compare it to a northern shore? We have to do it by lots of exploration. And so I would talk a whole lot more about this, but as you can probably tell, I love talking about this. So please contact me in person about anything here. There's so much to talk about. So real quickly, I'll talk about what we're planning on doing with this work. I'll start on the right there, continued scientific study. There's lots that we can do. So practicing our methodology, we'd like to make a larger project in which we survey the entirety of the Keweenaw Peninsula across multiple different types of bedrocks and find the locations of specialized plant communities that are unique to that region. Along those lines, we also have lichens and mosses that also only grow on the Keweenaw Peninsula. We haven't even touched that. And lichens are, of anything, likely the most connected to their rocks since they have such an intimate relationship with them. They grow right on top of them and penetrate them with their hyphae. It's very cool. And then to the left there, of course, the thing that we want to translate all of this eventually into is place-based education. And we're just in the beginning steps of this. Our connections with Michigan Tech and the Keweenaw Community Forest Company, they are well versed in this type of education. And so what we need to do is better understand these shorelines so we can take people out and share all the reasons why there's something to love about them, why there's always something to connect with, whether it's the plants or the rocks or the views or the water or the sounds. There's always something to enjoy. And so we want to emphasize those by getting to understand these shorelines ourselves, because before we study any type of ecosystem, you have to know the ecosystem and you have to develop a relationship with it. Otherwise, you're missing the point of being a land steward. And real quickly too, we've already started doing this, even with our preliminary first work. We're planning on extending this for several years, but this year we got to practice with a graduate level course at Michigan Technological University, the Lake Superior Exploration course led by Dr. Amy Marcarelli. It's a 2-week field course. And we had the opportunity to guest lecture for them and take them out to one of these shorelines and tell them everything that I've told you about today in a much greater capacity, and show them cool rare plants and tell them about how those little fractures along the rock house special plants that will only grow there in the state of Michigan. It's really beautiful. And with that, I'm finished. If you guys have any questions, please feel free to email me.
That was awesome. Thank you. Your passion for this work really comes through. Can you tell us a little bit more about what opportunities you might have to continue to engage with this project or help analyze the data? Like, will you get to be a part of that?
Oh, absolutely. A lot of the work that we've done here, the Keweenaw Community Forest Company and my partners at Michigan Tech have been really inclusive and letting me kind of run wild with some ideas. And I feel a great deal of gratitude, but I have a lot of stake in this work too. A lot of this conceptual work is stuff that I've been talking with the geologists at Michigan Tech with for almost a year now. And so it feels like a very personal project. And I really look forward to working with them because many of them specialize in creating field trips for educators and working with kids directly in K through 12 during all types of year. They work directly, they're part of the Lake Superior Stewardship Initiative, which is part of the Great Lakes Stewardship Initiative. So there's immediately that connection there. The additional connections with conservationists that I've been working with on the Keweenaw, it's amazing to see the passion that they have. There's something about the Keweenaw that people love that, you know, I'm very lucky to feel like I've tapped into. And it makes me feel impassioned to continue all of this.
So looking at all those different shorelines area, what have you seen in terms of the negative impacts of human activity? Please tell me you didn't see graffiti and whatever like they're finding in the Grand Canyon and other places.
Yeah, so in general, the good thing about a lot of these shorelines is that there are select areas along the shores which are easy to access, but in general, they can be really challenging to get to and they're challenging to walk on. So some of the higher trafficked areas, I have seen a little bit of graffiti. And obviously the places where people walk a lot, especially the public parks, they tend to have a much lower species diversity as a whole growing on the rocks just from trampling. The good thing is that they make up a smaller portion of the shoreline. And so we're able to walk into some places that, you know, may only be a couple hundred yards from a very highly accessed place, but they tend to be less trafficked. And so they still house a lot of the rare plants and they aren't super disturbed. And working with some of the people who live on the Keweenaw too, in general, the shorelines are typically not super built upon. So there's not a ton of development on the shorelines themselves. Up to the shoreline, absolutely, but not the shoreline itself. So that's been a real positive that I've seen. And these places are very special. There's no ecosystem like it in the state of Michigan. So knowing that they're probably protected under state land, under conservancy land, even under a lot of private ownership is, it makes me optimistic.
I guess kind of like a follow-up question with that, with kind of the curriculum that you're working on, and especially again, incorporating some of that like place-based learning methodology. How are you kind of thinking of balancing the trade-offs of that place-based learning and getting people to get outdoors and experience the Keweenaw Peninsula while also trying to leave a little bit less of that human impact? Like, I guess what to use kind of that limit there of allowing people to experience it without, you know, as you said, kind of trampling things and leaving that footprint there.
Well, that's a good question. And that's a tough one too. The good thing about a lot of these ecosystems is that the access points that we do have right now, while they are heavily trafficked and there is some degradation of habitat, they are prime examples along the shorelines, even with those, even with some of that higher visitation, because we're not just talking about the plants, we're also talking about the geology. And so thankfully, the rocks themselves aren't being degraded right now. They're just walked upon. They don't really mind. And so that in that aspect, the geology portions of the place-based education is going to be is probably going to be focused around those easy access points. And then if we're to do some of those plant studies, those are going to have to be more fine-tuned. And that's something that we'll be developing in the next year.
All right, unless there are any final questions, I think we'll wrap up and head to the next speaker. Thank you very much.
So my name's Ellie. I'm going into my senior year at Lake Superior State University, And I'm looking at relationships between zooplankton and larval lake whitefish in shallow water habitats across the upper Great Lakes. As you can see, this little guy in the corner, this is a larval cragganine, and he's about 15 millimeters. First, I want to start off with my acknowledgements. Thank you to all these organizations that helped in some way on my project or with the larger project that I'm working on. And a big thank you to Michigan Sea Grant for this summer internship opportunity and for funding my research for this summer. Also, I want to thank the Michael Fraker Student Memorial Fund for providing the travel funds. With that fund, I got to go to a big conference in Madison, Wisconsin for the Association for Sciences of Limnology and Oceanography. And I got to present my research that I'm presenting today on a poster there for scientists from all around the world. First, starting with the background of the lake whitefish. They are a key species to the upper Great Lakes, but the populations have been declining recently in some areas of the upper Great Lakes. Looking at this picture that I have from the Michigan Sea Grant website, you can see the life cycle of the lake whitefish. So they spawn in the fall and they lay their eggs close to the shoreline. And then the ice covers them up. They incubate there all winter, and then when the ice comes off the Great Lakes, they hatch, and then when they're sac fry and larval stage, they stay close to these shore water habitats, which is their larval stage is what I'm going to be looking at for my project. And then when they go to the juvenile stage, that's when they go off further into shore, and then they're not in the near shore habitats anymore. So looking at their food web changes, and especially in Lake Michigan, Lake Huron, the zebra and quagga mussels have invaded those Great Lakes, and they have impacted the food web. So these mussels filter out the phytoplankton in the water column, which is in this bottom area right here. And that's the food resource for these zooplankton. And the zooplankton is what these larval fish eat. So basically they're starting at the bottom of the food web, taking out the phytoplankton, which is less food for the zooplankton, so there's less zooplankton, so there's less food for these larval fish. And not just these larval lake fish eat these zooplankton in this larval stage, all of these larval fish do, so they're also competing with all these other larval fish for the same food resource. And then these larval lake whitefish depend on these zooplankton because these zooplankton are in their larval habitat, which is the nursery for these fish. So just to further understand why they're declining, we're studying their larval stage and their food resources available at this stage to see if this is one of the stages they're not making it to this older stages. Looking more at the zooplankton ecology, zooplankton and larval fish can exhibit these daily, vertical, and horizontal migration from day to night. And so these zooplankton dynamics can affect where these larval fish are found. So in this picture right here, during the day, these zooplankton go down further into the water column because they are harder to see because it's darker down there. And then at night, they come up closer to the water surface because they're still using that moonlight and the darkness to hide from these predators while also foraging for food. Zooplankton and larval lake whitefish are mostly collected at day in these deeper waters. And for my project, I'm looking at these nearshore habitats. So there's less data in these shallow water habitats. So my research is focusing on these areas that we know less about. So we need more data to characterize these zooplankton dynamics and changes over time in these shallow water habitats, which is where these larval fish are found. And then, so for example, larval lake whitefish at this larval stage, and with research and studies that other people have done, they estimate that these larval fish need about 20 zooplankton per liter of water in order to survive and grow into these older stages. So that brings me to my research questions. I'm looking at if zooplankton density and biomass and larval lake whitefish abundance differs between day and night and on the beach and near shore depths. So for my beach, beach is considered anything from 0 to 1 meter depth, and those are towed in waders and we pull the nets. And then for near shore is anything between 1 and 10 meter depth, and that is done off a boat. And I'm also looking at if relationships exist between zooplankton density and biomass and larval fish abundances. So seeing if how much zooplankton in the water column affects how many fish are in the water column. And to study this, I'm focusing on this little red rectangle right here is where all my data come from. But these other dots on the map are collected by these agencies on the bottom in 2023 as a larger project that's funded by the Great Lakes Fisheries Trust. And so for my sampling locations, we sampled about 20 sites, and each site was sampled on the beach, but only a few of these select beach samples had paired near shore samples from the boat, and each one was sampled night and day. So we had some really long nights and some really early mornings because we sampled dawn, dusk, night, and day. So we have all those variables. And each site was sampled up to 3 times every 2 weeks, starting ice off in 2024. So this year we started in late April and we went until like mid-June. Because these other agencies collecting their data, we had to meet up with them and organize a time for them to drop off their samples that they have collected. We worked closely with Bay Mills Indian community and closely with Sault Ste. Marie Tribe of Chippewa Indians to collect data in this square because a lot of teams for 20 sites sampling the same 24 hours. We need a lot of people. So for my sampling protocol, we did site characteristics at every site. We used a HydroLab Mini-Song, which is a picture of this in the top corner, to collect environmental data like water temperature, pH, conductivity, and DO, or dissolved oxygen in the water. We also collected chlorophyll A, so we took 1 liter of water out in the field, and then within 24 hours, we filtered 500 milliliters of that water with the filters in the top corner to be processed later. And then we also collected zooplankton larval fish. This net right here was used to collect zooplankton, and this is me and my coworker pulling the Newson net through the water. Both of these nets, they just were towed horizontally through the water column, just subsurface. The Newson net we pulled for 20 meters, and the zooplankton net we pulled for 30 meters. Each of the zooplankton and the larval fish that we collected were both preserved in ethanol to be processed later in the labs. And for fish, we had a tray at the end. We dumped all the contents into that tray. We collected 25 larval lake whitefish, and we counted the rest and we put them back into the water. But we kept 25 for the larger project because we're doing diet studies on those fish. So out in the field, we ID'd the fish down to cragganines the best we could. It's harder out in the field to identify these larval fish because they're so small. So then after that, we are sending them to USGS for genetic analysis to get down to species. So we're going to see if they're like whitefish or Cisco. This is also a picture of me and my coworker doing diets on these larval fish. So for my preliminary results for my statistical analysis, I did all my stats in RStudio. I ran a 2-way ANOVA to test for differences in zooplankton density and biomass at day versus night, beaches near shore, and their interaction with each other. I ran a t-test to test for differences in larval cragganines, catch per unit effort, which is CPUE, at day versus night on the beach. And I also used a linear regression to test for relations between environmental variables in zooplankton density and biomass, and to test for relations between zooplankton density and biomass and larval cragganine catch per unit effort. So for my results, we found that zooplankton density and biomass were both higher at night. This is zooplankton density was about twice as high at night as it was during the day. And then for zooplankton biomass, it was 3 times higher at night than it was during day. And about 80% of the zooplankton that we caught were copepods. So they were either Napolii, Copepodites, Dicyclops, which is this one up in the top corner, or Leptodioptimus. Since for running the 2-way ANOVA test, we found that there was the interaction between the day and night and the beach and near shore was not significant. So we're looking at these variables separately. zooplankton density and biomass were both higher, multiple times higher on the beach than the near shore. This one is also density. And then this one is the biomass. So you can tell that both of these were higher on the beach than on the near shore. And so far we don't have we also took vertical tows on the boat to get the full water column sample, which are not included in this data, but just looking at those, they were comparable to these beach zooplankton biomass and density. So for creatinine count was also higher at night versus day. So it was about twice as high at night than it was during the day, which is what the zooplankton were. So we're kind of seeing that the fish are following where the zooplankton are. And in this picture, this is what we dumped out of the Noosa net. So there are little fish in here somewhere, but we had to go find them and pipette them out of these trays and put them into their into a liter. We found that there was no relation between zooplankton density and biomass and larval carrageenan abundance. So this is zooplankton density. As you can see, there's really no relationship between them. And then also for biomass, there's no relationship, which is kind of interesting to see because we saw that larval carrageenan and zooplankton were both more abundant at night than it was during the day. But this shows us that there is really no relationship between them. We found that there's a negative relationship between conductivity and zooplankton biomass. But as of right now, no other environmental data related to these zooplankton estimates. But we have not looked at chlorophyll A samples yet. They have been processed, but they have not been yet included in this analysis. That's one of the things that we're going to look at further down the road to see if that was a relationship between them or not. So some of my ecological takeaways are that zooplankton larval fish may be higher at night because of migration patterns and the sampling protocol. So when we're sampling during the day, we're doing these horizontal tows just subsurface, and all those zooplankton are further down in the water column. So we're missing all of that zooplankton community and population. Also, zooplankton may be higher on the beach than the near shore because we're missing more of the water column than the near shore because that's anything between 1 and 10 meter depth. So these nets aren't covering the whole water column. We also took the vertical tows, so we're going to be looking at that at a later date to see if that makes a difference with the zooplankton populations. And zooplankton larval carrageenan abundance had no relationship. That probably is because of active consumption of zooplankton by these larval lake whitefish confounds any relationship between them. So when we get there and we sample, the zooplankton population and community could be different from what it was 2 hours ago because they're getting actively eaten during the day and during the night. So for my next steps, we're going to process the remainder of the zooplankton and fish samples that we received from other agencies that we got in 2024, and to examine more specific groups of zooplankton like Cyclopodia and Calanoidea. And then to incorporate the fish genetic data to species, the fish that we sent to USGS. So far, we've sent 600 fish to them. We should be receiving the genetic analysis back from them pretty soon. So we can go back into our data sheet and actually put if they're like whitefish or Cisco or what kind of fish they were. We're also going to do a more in-depth analysis to test for relationships between environmental data, zooplankton, and fish. So to wrap this all up, lake whitefish are important fish, but have been experiencing declines in the upper Great Lakes. Bottleneck for their survival could be at the larval stage. And so we need a better understanding of the stage using standardized sampling, and an ecosystem approach sampling many variables across space and time may help us better understand this problem. And with that, I'd like to thank you guys all for being here, and I'll take any questions or my email's down here if you guys want to email with questions.
I've got a question to start with.
Yeah.
That sounds like a lot of intense field work.
It was
and
very
a
intense.
great experience, but do you see yourself in a career that involves field work like that? Did this light a fire for you?
Yeah, the field work is probably my most enjoyable part. Even with the late nights. I think the one night we got back at like 4 a.m. It was pretty crazy. And then going out at like 6 a.m. But like doing all this data, collecting, pulling the tow nets, pulling the zooplankton nets, and just collecting all this data and then getting back to go process the same data that you just collected, I think was pretty cool. I can definitely see myself doing a lot more field work in the future.
A comment and a question for you.
Yeah.
So your relationship, although you say there's no relationship, it almost looks bimodal, especially the first one. And so my suggestion to you is as you're getting more data to overlay on those graphs, try overlaying time, try overlaying the, location that you collected it from. And then as you get the zooplankton group, overlaying that too. So especially in the first one, it does look like there are two relationships. They're just on the same graph.
Yeah. Yeah, definitely looking more at this environmental data with the zooplankton and the larval fish separately. Just with the time on this is
Oh, yeah.
what we had time for.
Yeah, like that's awesome.
But yeah, we have a lot more data to look at, and we have even more data to process for these 2024 sampling
And-
year.
So my question is, are you going to be part of that? Are you going to continue working on this your senior year, or what's your plan?
Yes, I actually just got hired back on to work on this project in the fall. So we'll be continuing to process this data throughout, well, in the fall and then probably in the spring semester as well.
I just had a question. So going back to that graph with the zooplankton density and the conductivity, I was just wondering if you had an idea of what might be causing that relationship, and if that is that related to turbidity, or I was curious if you had an idea of why that might be an environmental factor affecting it.
Yeah, right now we're kind of taking this as like a grain of salt, because we do have this dot way up there, and then these three over here that are kind of a little crazier. But other than that, that's just what we figured out. But if we even tried to remove these data points, and there was still like a negative relationship between these two variables.
Alrighty. So hi everyone, my name is Jake Downey and I am heading into my senior year at the University of Michigan. And I've been working this summer with the Michigan Department of Natural Resources Fisheries Division located out in Charlevoix. And our project was looking at the impacts of fishery independent data on Chinook salmon growth models. So first, to start us off, just want to talk a bit about why we're looking at Chinook salmon in particular. So for those who don't know, Chinook salmon aren't actually native to the Great Lakes. They have been introduced, originally being from the Pacific Ocean, they've been introduced since 1967 in an effort to control invasive alewives. So these fish since then have really taken off, especially in Lake Michigan, and are now one of the more dominant predators within the lake. And in addition, they have fostered a multi-billion-dollar sport fishery industry within Lake Michigan. And so these fish are essential to both the ecology and economics of Lake Michigan, making them highly important to understand from the side of managers such as the MDNR. And so nearly all of the data that we've collected on these Chinook salmon come from what's known as creel data, which is essentially approaching anglers who are catching these Chinook salmon and taking samples from them and gathering data from them. And so it is this kind of source of, again, mentioning that like participatory science of approaching these anglers and gathering a lot of what we use from them. So now my project has focused on some of the aspects and limitations of this creel data. And so while these creel programs are essential for gathering huge amounts of data, far more than the Michigan DNR could ever think of getting from independent surveys or even just partnering with other organizations, although we are able to gather a lot more of that data from this creel data, it may have this selective bias towards larger fish. If you think about it, when an angler goes out, a lot of times they are seeking out a lot of those larger fish. And even the gear that they use, like you can see things like our lures that a fisherman might be using over here. And even just the fact that there are these size and bag limits, all of this kind of leads to only the largest fish from especially older ages being selected for by these anglers. And so the fact that they're using these bigger lures that can't catch a small 5-inch newly hatched fish, and the fact that you can only keep so many of them and you have to toss back smaller ones, all of this leads to only large fish, or predominantly large fish, being selected for by our creel data. And so this means that there's very, very little known about younger, smaller fish. And so there's this big open gap when we're only using this creel data for what these younger fish are looking like within the real population. And a lot of the growth models that we use to assess how these fish are doing and to see if they're growing to these bigger sizes because they have enough food, a lot of those rely on accurate data throughout the entire age range for these fish. And so if we're missing some of that younger ages, it can be devastating to what we're doing within, or it can be misinforming kind of what we are choosing to do with our data. So this leads us to my question, which is, will the incorporation of reliable fishery-independent data into Chinook salmon growth models create a more accurate model that can better inform Chinook stocking numbers? And then as sort of a sub-question to this, just trying to tease out some of the differences between this fishery-independent data and our fishery-dependent creel data. So to start with, where we're actually getting our fishery-independent data from, this brings us to the site that I've been working with this entire summer, and that's the Ludington Pump Storage Plant based out of Ludington, Michigan. So now this is a hydroelectric facility that essentially pumps water uphill during low energy demand times, and then when people have their ACs on there and they're using a lot of power, they pump it back down so they can sell it off for a higher price. And so they need to use these big intake pipes to pump all of that water uphill. And so to keep fish from getting sucked up into a random reservoir above, they are legally mandated to set up these big barrier nets that allow them to hopefully keep fish out. So you can see that in this diagram right here, this big yellow line. This net is around 2.5 miles long, and it's actually the longest of its kind in the world. And in addition to keeping this net set out, they also are required to sample from inside and outside of the net to make sure that it's actually working, because what good is a giant net if fish are still getting sucked up into some of these pipes? So they sample from April 15th to October 15th, twice a week, to ensure that fish are not getting inside of the net. And they use these bottom set gill nets, which are, you can see these right here. They essentially just lay out these big old nets that they can use to catch fish. And these are a lot more generally selective than an angler's gear would be. And so instead of, again, these big lures that they're using to catch larger fish, these gill nets have mesh sizes that can catch fish as small as 5 inches up to a big 40-inch fish. And so they're very, very versatile and can catch a wide variety of fish, including a lot of our Chinook salmon that exist in the lake. And so they have been consistently sending us scale samples and biodata since 2018 from Chinook salmon. And we believe that this source of data might act as a really, really lucrative source of this, again, fishery independent data. So my work has been kind of looking at all of these scale samples and bio sending us for the past 5 years. So I mentioned scale samples, and the reason that we're looking at those in particular is that much like trees, these scales develop distinctive rings that we can use to count the ages of our fish. So you can see on our helpful diagram here that we have these small individual little lines that build up over time. Those are known as circuli, and those are just the tiny little layers that grow over time. But then we are really focusing on are these darker bands that you see here. And so those are known as annuli and are clusterings of our small little circuli lines that develop in the winter. So essentially, when it's wintertime, the fish is sluggish, it's not eating as much. And so it can't put that energy towards growing its scales. And so we get these really, really distinctive dark bands that are these clustering of circuli banding together. A unique thing about salmon is that because they are hatched within streams and then swim down to the lake when they mature or when they begin to develop, they form this little false annulus right in the beginning here. And that's essentially when they're making that taxing and journey downstream and they're not eating as much when they're making that journey. You get this little clustering of a band at the very beginning. So just a unique little kind of ripple and kind of how we look at salmon scales. But so we take all of these scale samples that we get and we press them onto acetate plates using basically looks like a miniature version of like an industrial roller. And so we press them onto these plates so that when I age them and I say this fish is 1 year old, I can give it to another person who works for the fisheries department and they can say, yes, that's 1 year old, or actually I disagree, that's 2 years old. And so we can validate those ages and also make sure that when we do validate them, that we're using the same samples of scales. And so it keeps our data consistent. Once we press them, we are reading them under a microfiche reader, which is one of these guys right here. So this was my first time really encountering one of these. And folks out here who are around my age, we might not be super familiar with them, but I was vaguely familiar from seeing things like Stranger Things and like other 80s content where you kind of see some more of these guys. So yeah, using actually some of this relatively older technology because it's super clear to see what these scales look like here. So I was working with those this whole summer. So now diving into some of the modeling and statistical tests that we were doing. So we were modeling all of our growth estimates using what's known as von Bertalanffy growth models. This is basically the quintessential way that we model growth for all kinds of fish species. And essentially what we are trying to estimate is the length at time t. So trying to figure out when a fish is 1.5 years old, about how long is it going to be. And in order to estimate this, we need to also estimate several parameters for that growth. And that includes this L infinity right here, which is known as our asymptotic average length. Essentially, it just means what's the maximum length that this fish can reach during its lifetime. We also need to know the growth rate coefficient, K, which is essentially how quickly is this fish growing over time. And then finally, T naught, which is the theoretical time at length 0, which is kind of like trying to figure out some of our lower bound estimates. And so all of these parameters are oftentimes estimated from the population that you're looking at. And in order to accurately estimate those parameters, you need to know accurately what each life stage of that fish looks like. So again, going back to the fact that if we have these gaps in the creel data, we may not be getting an accurate idea of what some of these parameters look like. And so we're hoping to fill in some of the holes in that with our fishery independent data. And so to look at this from a statistical perspective, we were using something called a likelihood ratio test. This is also a common way to compare our von Bertalanffy growth models. And essentially it's just saying, whether or not we can hold our different parameters equal for each of our different kinds of data, in this case, our fishery independent and fishery dependent data. And so we have our different alternative hypotheses that each hold different parameters equal. And then our null hypothesis in this case would be stating that all of our parameters are completely different. Our dependent data would be completely different from our independent data. And then we'll look at some significant p-values which indicate that we prefer this null hypothesis that our data is completely different from each other. So now diving into some of the results that we were to get. This is a violin plot where some of our wider little plots here indicate that there is a higher density of that data. And so the important thing to notice here is that we do in fact see a big difference in our younger fish. So these are fish up to age 1, so we call them our age 0 fish. And we see that there is a noticeable decrease in the lengths of our fish from our fishery independent Ludington data. Now this means that, again, our creel data, when we're only catching the largest individual or the larger individuals at our younger ages that are actually able to grab onto the gear that people are using, we're only going to be representing the very peak, basically the statistical outliers of our age 0 fish. And so when we use these gill nets that can less selectively collect our different fish, we're able to see a more probably accurate estimate of what these smaller, younger fish are going to look like. However, we have a bit of an opposite problem for our older fish, because older fish generally are more rare within the ecosystem. And so while our creel data, just because of its sheer size and the fact that they are trying to get these older, bigger fish, they are able to get a lot more data from these larger, older fish. However, our Ludington data is only representing one of these 4-year-old fish. And so we do see kind of these opposite problems from different sides of the data here. And then there was a weird kind of wrinkle here where we see this odd bimodal distribution within our age 1s. And so we were a little concerned about what might be causing this. So we first looked at sort of a seasonal relationship that might be occurring. And we do see that there is actually a distinct increase within the sizes of our age 1 fish from our Ludington data in the fall. In this case, we categorize the fall as any time after August 15th. In this case, our spring and summer would be from April to August 15th. And so we do see this weird relationship where in the spring and summer, our Ludington data, as we would kind of expect, has smaller age 1 fish compared to our creel data. But during the fall, it actually has larger fish than our creel data, which is this weird sort of wrinkle that we didn't know how to parse through. But we believe that this is likely because the area around the Ludington pump station is actually a common spawning ground for Chinook salmon. So we believe in addition, spawning fish are also drawn in by the sounds of rushing water because they're trying to swim up those streams. So we believe that mature age 1 fish, which if an age 1 fish is already ready to spawn, that means that they're likely very, very big for their age. These mature age 1 fish are coming back to Ludington, drawn in by the sound of these rushing water from the intake pipes, and could potentially just be caught outside of the net or even likely be jumping over the net to try and get to those the sound of that water. And so this is a concern because, first of all, we want to be using these fish caught inside the net as little as possible, because if a fish is able to jump over the net or swim through the net because they're small enough, that could be biasing some of the sizes that can get through. So that would be some bias data there. And in addition, we want to make sure that if it's only these mature fish that are being selected for out there, that could be biasing the data again to these larger age 1 fish if it's only mature ones that are coming back. So because of these 2 wrinkles there, we decided to exclude all of this data from our fall set. And so anything after August 15th was excluded from our growth data just for the sake of looking at what we believe to be a truly mixed population. So this is what we're left with here. We do still see that even with excluding that fall data, we do get a lot of really, really good data from our age 0s in our Ludington data. And we do also still get some small age 1s that could fill in that gap over there. So now looking at some of our statistical tests. Again, this is from our likelihood ratio test. And the big significant value to look at here is this one for this H0 versus H4 test. In this case, what this means is that our null hypothesis stating that our two types of data are completely different from one another would be preferred over one stating that they're completely the same. So now we can't rule with this where that difference lies, but we do know that there is some gap in our fishery dependent data that is being filled in by our fishery independent data. There's some difference between the 2 types of data that is giving us this significant value. And we can see that in our graph here. This is the actual von Bertalanffy curves for each of them. Again, our blue is our Ludington data, our red is our creel data. And we can still see, again, because of all of those smaller age 0 fish, that it drags that bottom part of the line down a little bit, again, likely indicating a more accurate relationship between for younger fish. However, our opposite problem, again, that our older fish have a line that's higher up because we're not getting those older fish to kind of tamper that line down a little bit. And so we kind of have opposite problems here where our Ludington data is filling in a lot of the gaps for our younger fish, but our creel data is filling in a lot of the gaps for those older fish. So kind of summarizing this as a verdict on our fishery independent data, again, as we kind of predicted, we do get a much better understanding for these smaller, younger fish from our fishery independent data. We're catching things that just simply cannot be caught with the gear that anglers are using. And so we're able to actually get accurate data, we hope, for these younger fish. We hope that this will lead to better parameter estimates, particularly we predict for that T0 value, because again, that's kind of predicting that length at age 0. And so you need to know accurate data for those younger fish. And in general, we just believe that this sort of fishery independent data can help fill in those biases created by creel, by the kinds of fish that they're able to catch. However, places such as the Ludington Pump Storage Plant are incredibly rare, and operations like that tend to be very, very expensive. And so finding actual sources of fishery independent data is difficult. And that is a large reason why we likely don't capture those older fish, because there just simply isn't enough data out there right now to collect them. And so because of that, it's difficult to capture, again, that right side of that graph, that asymptotic length. We can't really predict where that's going to cap out at if we don't catch those larger fish. And so going back to the fact that these 2 kind of complement each other, we believe that the best case here is to combine our fishery independent and dependent data. they both are able to complement each other well, with one fleshing out, again, that left side of the graph, the other that right side. And they seem to work really, really well together to create this nice marriage of the two data sets. And so that is kind of our big idea going forward, is to continue to pursue this fishery independent data and combine it with the creel data that we already have. So speaking of those next steps going forward, this was actually the first time that we've looked at this Chinook data from the Ludington set, and really that anything was done with the Ludington data so far. And so kind of dipping our toes into this for the first time, there was a lot that we learned in terms of the protocol that we want to work on changing. This includes just getting better clarification on the procedures. Because we're not the ones out there collecting the data, it's very important that we know exactly what the folks at the pump storage plant are doing to collect this data. And so we just need to make sure that there's better communication between us and them so that we know what's going on. We also want to get better labeling for what fish are caught inside of that barrier net. Again, if only fish that are mature and willing to jump over the net, or fish that are small enough to wiggle through, are going to get caught inside of the net, that is a clear bias in some of our estimates there. And so we need to do all that we can to make sure that we can limit those from our data set to make it a better representation of what the community looks like. And then finally, we do want to know the maturity of these fish, again, because if those biases might exist for these more mature fish coming during the fall, that would be very useful to really put a button on that and say that that's where that lies. We also want to start incorporating this into some predator-prey models. Again, we started this off by saying that Chinook salmon are introduced to understand to predate upon invasive alewives, and so the relationship between alewives and Chinook salmon is now very, very important for understanding how the populations will change over time. And so we hope to incorporate our biodata that we got, in addition to some catch data that we've been preliminarily looking at. Again, going back to the catch per effort idea that we were looking at with the last presentation. So using some of those to understand how the populations shift over time. Then we also just want to encourage and talk to managers about creating more sources of this fishery independent data. We believe that it's really, really valuable for filling in some of those biases of creel. And we've already heard with some preliminary talks with managers, a lot of excitement towards creating more kinds of this data and filling in some of the gaps in what we have. And so we hope that there might be opportunities to either find sites like Ludington or to establish long-term surveys ourselves where we can just set nets down and routinely pull them so we can get consistent data from one location, which could be really, really valuable. And along the way, I've just been helping out where I can. I've been very, very fortunate to help out and attend a lot of different kinds of field work at the Charlevoix Fisheries Research Station, whether that be pulling up gill nets to look at fish community surveys or helping to try and catch sturgeon in places. Or this was a acoustic survey that was done actually right in Ludington. And so the pump storage plant is actually right behind me in that photo, even though it's a little bit far away. But a lot of incredible opportunities that I have immense thanks to all of the folks at the Charlevoix Fisheries Station for allowing me to kind of partake on and to learn a lot from them. And with that, I do want to acknowledge all of them, in addition to Sea Grant for funding and sponsoring this project, the Michigan DNR for taking me on, and for all of the wonderful folks there, especially Ben Tershak, my research mentor, who has been guiding me through all this data and has been with me every step of the way to make sure that this has come to fruition. And then all the other folks who have helped me kind of clean up this data, analyze all of it, and take this project to where it is now. Yeah, so thank you so much for listening, and then thanks to all the folks who have guided me along this path.
I'm Ben Tershak. I'm with Michigan DNR. I was working with Jake over the summer. It was I just want to, this is more of a comment than a question, I guess, but I really wanted to thank him for all of his work. This is you know, we spend an awful lot of time thinking about and attempting to model and understand these populations in Lake Michigan, understand their population dynamics, and sort of we had this data source that really hadn't been looked at in a long time, and Jake really, like, you know, sort of grabbed this bull by the horns and gave us an opportunity to look at an entirely different data set and gave us a much different perspective than what we've had in the past about the growth and size and age of salmon. And I think what might not have been really clear from his talk was that sort of really changes and identifies like biases in our population models, in particular how much these things have to consume to grow. So if they're much smaller at age than what we've than what we previously thought from our fishery dependent creel monitoring. That means they must have eaten a lot more to grow from that, you know, size at age 0 or age 1 to size at age 1 or age 2, which means that, you know, there's a lot more consumption, which means that there must be more prey out there than what we, you know, had previously thought. So this is, it really just emphasizes sort of the importance of these data in understanding predator prey dynamics, which is sort of a linchpin to management in Lake Michigan. So I just wanted to sort of applaud him for that effort and you know, recognize how meaningful this will be for management moving forward, I think.
Thank you. It's very encouraging to hear how these projects fit into the broader paradigm of fishery management. So we're glad to hear that.
Yeah, great presentation. My question is, was there a reason to use the data from inside the net? Because just as a little context, I was a trustee in the Great Lakes Fisheries Trust till recently. We spent a lot of time looking at the data of what's outside the net versus inside to look for, well, the differences, right? To see if the net was working. And then the assumption then was if it was inside the net, essentially it was going to get chewed up by the Ludington Pumped Storage, which is what provides the funding for the Great Lakes Fisheries Trust in mitigation. But so I'm aware that there's very big differences in data between that. That's the whole goal. But it sounds like you use both inside and outside data. And I was wondering why both and not just the outside.
Yeah. So again, this goes back to communicating with the folks at Ludington, and that's the fact that they actually didn't tell us what was what. And so all we have is the scale card for what the fish was caught. We have the catch data saying that we caught this many fish inside and outside on this day, but we have no way to assign, okay, this fish, let's say like this 20-inch fish was caught here. We have no way to assign whether that was outside or inside the net. So again, that goes back to things we want to clear up with them and be like, hey, we need to know that for our kinds of analyses. But because this was the first time that we've used this data, they never told us that. And so there was no way to know. So unfortunately right now, everything that we do look at is both inside and outside. That's why we limited it to only the spring and summer, because we did find that the majority of the fish that were inside the net were caught during the fall and were predominantly larger. Again, we think it is those larger mature fish that are desperately trying to jump that net to get to what they think is their spawning ground. So yeah, limiting it to just the spring and summer was an effort to control for that. But as of right now, there was no way that we could know. So that's something we want to enforce in the future.
Thank you.
Great presentation. The first question that I have is this problem just in the Great Lakes, or has there been similar work in other bodies of water, like testing between creel data, independent data, and kind of fusing the two like you're aiming to do that you could kind of model your work after?
Not that I'm aware of right now. Especially with Chinook, there have been past attempts to implement some fishery independent surveys. The DNR and other agencies would essentially set up gill nets that they would sit on for a night, then pull them up, but they were incredibly variable, and it was very boom or bust, where they would either pull up nothing or they would hit a school of fish and you would pull up hundreds of Chinook salmon at one time. So while they got good biodata from that, when you're trying to do, again, some of this population modeling that I'm hoping to kind of get towards in the next steps, for that, you need to, again, know those accurate like catch data. And if it's either you're catching a lot or you're catching nothing, that's not a really good way to look at actual catch per effort cycles. And so that survey was discontinued just because it was so boom and bust. But outside of that, yeah, especially with Chinook, it's been predominantly creel data. And I think a lot of fish on the Great Lakes are predominantly creel just because it's such a large source of data.
I think we are ready to wrap up then. So thank you very much.
Thank you guys.
And we'll do a thank you to our online audience for joining us. Thank you for your participation today, whether you're Michigan Sea Grant staff or connected to one of the speakers today. We're grateful, and we are going to, and the mentors and the sponsors. You're grateful for all that you've done for the students that we were able to hear from today, and hope to continue these partnerships into the future. So we will bid you farewell.